Have you ever paused to think about how quickly our global scientific community can rally against a new health threat? Or perhaps how your annual flu shot keeps transforming almost magically to stay one crucial step ahead of mutating viruses?
It's a profound question, isn't it? Really touches on everything from our daily health to, well, global well-being.
Exactly. And it brings us right to the heart of what we're diving into today.
Indeed. We're about to embark on an in-depth exploration of the world of vaccination, and it's a field that's far more dynamic and multifaceted than most people probably realize.
Right.
It's not simply a medical procedure. It's this vibrant, often really complex interplay of groundbreaking scientific innovation, deeply considered ethical decision-making, the ever-shifting tides of public perception, and even the high-stakes chess game of global strategy.
Wow.
It's truly a microcosm of so many forces at play in our modern world.
Okay, let's unpack this intricate topic then. For this deep dive, we've pulled together a substantial stack of diverse sources. We're talking everything from cutting-edge scientific journals detailing the latest breakthroughs in mRNA cancer vaccines and the future trajectory of vaccine development,
Yeah, some really fascinating stuff in there.
to analyses of social media rhetoric, which is a whole world in itself, and critical reviews of public health policies and their ethical underpinnings.
Right, the policy angle is crucial.
So our mission today is basically to distill all of this into the most important nuggets of knowledge, giving you a real shortcut to being truly well-informed. We want to uncover those surprising facts and deliver those genuine aha moments that make complex topics click into place.
Hopefully, yeah. Got through some of the noise.
So get ready, because we're going to dive headfirst into the incredible science that's revolutionizing prevention, delve into the profound societal impact of these medical marvels, and gaze into the exciting, sometimes challenging future of how we protect ourselves and our communities.
Sounds good. Where should we start?
So let's kick things off with the incredible science that makes all of this possible. When many of us think about vaccines, our minds might still go to the traditional methods, the ones that have been around for a long time, the tried and true.
Yeah, the old school ways.
Exactly. But how exactly did those older methods work? Can you walk us through that?
Sure, that's a great place to start because understanding the past really helps us appreciate the present revolution. For decades, the traditional approach was, as you said, quite old school. Picture scientists essentially acting as cultivators.
Yeah, okay.
They would literally grow viruses or specific pieces of viruses, often proteins, in enormous vats of cells. Think of it like a giant biofarm for viral components.
Ah,
A classic example that many people are familiar with is how most flu shots were, and some still are, famously produced in fertilized chicken eggs.
right. I remember hearing about that. Chicken eggs.
Yeah. Once these viral components were grown in sufficient quantities, they'd then be carefully purified. A small, inactivated, or maybe attenuated, weakened dose of these purified components would then be injected into you as a vaccine.
So it's the actual virus bits, but made safe.
Exactly. The idea was simple, but effective. This dose trains your body to recognize the infection. So when the real live pathogen eventually arrives, your immune system is already primed and ready to fight it off. It often prevents illness altogether, or at least significantly reduces its severity.
That makes perfect sense. Your body learns to recognize the mugshot of the virus without ever having to face the full criminal, so to speak.
Right, good analogy.
But I imagine growing viruses in chicken eggs or massive vats of cells.
No. Well, it sounds like a lengthy process. Is that why it could take so long to make vaccines that way?
Precisely. That's the big drawback. The significant disadvantage of this traditional method is indeed its considerable timeline.
Okay.
The cultivation, the purification, all the quality control steps. They're inherently time-consuming, often taking many months. Months.
Wow.
And this slow pace became a glaring vulnerability, particularly when faced with rapidly emerging health crises like new flu strains or, novel pathogens like we saw with COVID. The world needed a faster, more agile way to produce vaccines at scale.
And that's where mRNA technology dramatically entered the scene, fundamentally changing the game. This isn't just an incremental improvement, is it? It sounds like a true revolution.
Oh, absolutely. It's often described as a paradigm shift, and that's not an exaggeration.
The M in mRNA stands for messenger, right? And it really lives up to that name. It sounds almost like a biological instruction manual.
It absolutely does live up to its name, and the instruction manual analogy is spot on. Messenger RNA, or mRNA, is a naturally occurring molecule in all our bodies.
Okay.
Its fundamental role is to carry genetic instructions, the messages from our DNA, which is safe in the nucleus, out to the ribosomes in the cell's cytoplasm. The ribosomes are essentially the cell's protein-making factories.
Scientists figured out how to basically harness this natural process. They learned how to create these messenger RNA molecules in a lab. So instead of injecting the actual virus or its proteins into you,
Don't inject the instructions.
exactly, you're injecting the instructions, the blueprint for your own cells to make a piece of that protein. It's like giving your body a recipe rather than giving it the cooked meal.
So your body essentially becomes its own mini vaccine factory, which is just mind-blowing when you think about it.
It really is quite elegant.
You get this tiny snippet of genetic code, your cells read it, they produce a specific target protein that mimics a part of the pathogen, and then your immune system recognizes it and builds a robust defense, all without ever encountering the actual virus itself.
That's the core idea.
Can you walk us through the actual process our cells go through when they receive those mRNA instructions? And a key question many people had. Does that genetic code stick around in our bodies? Does it become part of us?
Great question. And it's a common point of misunderstanding. So when you receive an mRNA vaccine, the mRNA is encased in a protective lipid nanoparticle, basically a tiny fat bubble.
Okay, like a delivery vehicle.
Exactly. That helps it enter your cells. Once inside, those instructions are delivered to your ribosomes, the protein factories. Your cells then briefly use those instructions to produce the specific viral protein, often a harmless piece of it, like the spike protein of SARS-CoV-2.
Right.
This protein is then displayed on the surface of your cells, or sometimes it's released. Your immune system, acting as the body's vigilant surveillance system, recognizes this foreign protein.
Because it shouldn't be there.
Right. It then mounts a response, creating antibodies and specialized T cells that can remember and neutralize the real virus if you encounter it later. Now, critically, the mRNA itself is transient. It's temporary.
Okay, so it doesn't last.
No, it never enters the cell's nucleus where your DNA is stored. That's really important.
Okay.
And it degrades naturally within a few days or weeks, long before the immune response is even fully developed.
Phew, so it's a temporary message, not a permanent change to your genetic makeup.
Absolutely not. A temporary instruction set.
And the beauty of it is that it's a much, much faster process for developing and deploying vaccines. This speed proved to be incredibly crucial during the COVID-19 pandemic, didn't it?
Oh, massively.
It felt like these vaccines came online with just unprecedented swiftness.
The pandemic truly served as the ultimate, albeit tragic, proof point for mRNA technology. The rapid development and deployment of these so-called mRNA vaccines saved millions of lives during the COVID-19 crisis. It unequivocally demonstrated their capacity for rapid response in a global health emergency.
Millions of lives, that's staggering.
It is. Michael Osterholm, a widely respected expert on pandemic preparedness, really put this speed into perspective. He noted that with older vaccine technology, it might take 18 months just to vaccinate about a quarter of the world against a pandemic flu strain.
18 months for a quarter of the world.
Okay. But with mRNA technology, he stated it could change that dramatically, such that by the end of the first year, we could vaccinate the world.
Wow. From 18 months for a quarter to potentially 1 year for everyone.
That level of speed was previously unimaginable. It offers a truly revolutionary tool in our arsenal against fast-moving global threats.
And this isn't just some neat trick or a flash in the pan. This is Nobel Prize-winning innovation.
That's right.
In 2023, Doctors Katalin Kariko and Drew Weissman were awarded the Nobel Prize in Physiology or Medicine for their groundbreaking discoveries concerning nucleoside base modifications. Now, for listeners who might not be deep into molecular biology, could you explain why these modifications were so absolutely essential? What problem did they actually solve?
Yeah, their work was truly pivotal. See, initially, when scientists tried to introduce synthetic mRNA into cells, the body's immune system often perceived it as a foreign invader, almost like a viral infection.
Ah, okay.
Yeah.
It attacked the instructions.
Exactly. It would launch a strong inflammatory response that would quickly destroy the mRNA before it could even deliver its protein-making instructions. This was a major, major roadblock.
So the message wasn't getting through.
Right. What Keiko and Weissman discovered was that by subtly modifying the nucleoside bases, the actual building blocks of the RNA molecule, they could essentially disguise the synthetic mRNA.
Like putting a cloak on it.
Sort of, yeah. This allowed the mRNA to slip past the immune system's initial alarm bells, avoid being destroyed too early, and effectively deliver its message. This led to much more robust protein production and, crucially, a proper immune response against the target antigen without causing that harmful inflammation.
So without their discoveries.
The mRNA vaccines we now rely on simply would not have been possible. It was a true, fundamental scientific breakthrough that unlocked technology's potential.
That's a fantastic explanation. Makes it really clear how critical their work was. So what's fascinating here is that while the COVID-19 mRNA vaccines aren't perfect, we know protection does wane over time, and they are strongest against severe infection and death rather than stopping all transmission.
Right, they're not sterilizing immunity in most cases.
Their adaptability is a huge game changer. Both the coronavirus and flu viruses continually mutate, right? That's why we often need annual flu shots.
Exactly, they're constantly changing their disguises.
But mRNA vaccines can be updated much more quickly each year than traditional types. That seems like a massive advantage.
It's an enormous leap forward. The ability to rapidly redesign an mRNA sequence to target new variants or strains is huge. With traditional vaccines, you're often stuck in a months-long production cycle, trying to predict which strain will dominate next season.
Kind of like forecasting the weather, but for viruses.
Pretty much. And if your prediction is off or a new strain emerges rapidly, you're already behind the curve. With mRNA, you can theoretically take a new genetic sequence from a circulating virus, plug it into the existing mRNA platform, and scale up production in a fraction of the time.
So much more agile.
Exactly. This agility makes it a powerful tool for staying ahead of, or at least quickly catching up to, rapidly mutating threats like influenza and coronaviruses. And this versatility means the technology's impact extends far beyond just COVID-19.
Okay, here's where it gets really interesting and, quite frankly, incredibly exciting for me. This amazing mRNA platform, proven so effective and rapid for infectious diseases, is now moving into entirely new horizons. Scientists are using it to try and develop vaccines and treatments against a long list of diseases we've struggled with for ages, including really complex challenges like cancer and cystic fibrosis.
Yes, that's the next frontier.
This truly sounds like a profound shift in focus for the entire medical field.
It absolutely is a profound shift. The success of mRNA in infectious diseases laid a robust foundation, built that confidence and infrastructure. And now scientists are actively leveraging this technology to develop therapies and preventative measures against a wide array of other ailments.
And cancer specifically, I mean, that's huge.
For cancer specifically, this is being hailed by many as a new epoch in cancer immunotherapy.
A new epoch, wow.
Imagine teaching your own body's immune system to recognize and attack cancer cells. That's the core promise here. These mRNA cancer vaccines work by encoding tumor antigens.
Tumor antigens, what are those exactly?
They're essentially unique markers or proteins found on the surface of cancer cells, or sometimes inside them, that the immune system can learn to target. The mRNA tells your cells to make these markers, showing them to the immune system so it knows what to look for on the actual cancer cells.
So it's like putting up wanted posters for the cancer cells using the body's own communication system.
That's a great way to put it, yeah. Eliciting potent immune responses against the cancer itself.
That sounds like a dream come true for oncology. So beyond just the speed, what are the key advantages of using mRNA technology specifically for cancer treatment? What makes it such a strong candidate for this monumental challenge?
Well, there's several critical benefits that make mRNA uniquely suited for cancer. Firstly, as we touched on, it has an exceptional safety profile.
Right, no integrating into the DNA.
Exactly. Unlike some older gene therapies that might try to insert genetic material into a patient's own DNA, a process that carries a small risk of accidentally disrupting an important gene, causing what we call insertional mutagenesis.
Which sounds bad.
It can be, yes. mRNA vaccines don't do that. They deliver temporary instructions that stay outside the cell's nucleus, where our permanent DNA is housed, and they quickly degrade after delivering their message. This virtually eliminates that specific risk of altering your permanent genetic code.
Okay, that's a huge reassurance on the safety front. What about efficacy and personalization? Because cancer is so individual, right? So unique to each patient.
That's where mRNA really shines. It has the ability to concurrently encode multiple antigens.
Ah, so not just one target.
Right. It can present several different targets or wanted posters from the cancer cell to the immune system all at once. This significantly boosts the vaccine's effectiveness and, crucially, paves the way for truly personalized vaccines.
Personalized? How so?
Imagine sequencing a patient's specific tumor, identifying its unique markers, and then creating an mRNA vaccine tailored just for them. That's the goal.
Wow.
Furthermore, the transient nature of mRNA expression. The fact that it doesn't stick around forever ensures that the antigen stimulation is temporary. This is vital because it effectively reduces the likelihood of chronic inflammation or autoimmune responses, which can be a concern with therapies that constantly activate the immune system over long periods.
Okay, that makes sense. You want the attack, but not constant friendly fire.
Precisely.
And the speed advantage we talked about for infectious diseases, I assume that's still a major plus for cancer treatment, too.
Absolutely. The rapid design and synthesis of mRNA vaccines enables swift responses to evolving oncological challenges. This includes customizing vaccines to the unique genetic profile and tumor characteristics of individual patients, as we just discussed.
So if a tumor changes,
If a tumor mutates or becomes resistant to initial treatments, an mRNA vaccine can, in theory, be quickly redesigned to target those new vulnerabilities. That adaptability is key.
so it's safer, potentially more effective, highly personalizable, and still incredibly fast to adapt. It sounds almost too good to be true. I imagine there must have been immense hurdles bringing something this groundbreaking from the lab bench to an actual cancer patient. What were some of those critical challenges researchers had to overcome?
Oh, absolutely. You're right to point out the challenges. Early on, researchers faced significant hurdles, like the inherent instability of mRNA itself. It's quite a fragile molecule, easily degraded by enzymes in the body.
Right, it needed protection.
Exactly. Then there was a challenge of needing efficient and targeted delivery systems, those lipid nanoparticles we mentioned, to ensure the mRNA actually reached the right cells without being destroyed by the body's defenses first.
Get it past the gatekeepers.
Yep. And particularly in cancer, there's the big problem of immune tolerance or evasion by tumor cells. Cancer cells are notoriously sneaky. They can essentially hide from or even deactivate the immune system.
So making the immune system see them is tough.
Very tough.
Yeah.
However, the huge effort and success in developing and deploying mRNA vaccines for COVID-19 provided invaluable insights. It effectively helped overcome many of these technical bottlenecks in stability, delivery methods, and figuring out how to properly activate the immune system without causing harmful side effects.
So the pandemic response actually accelerated the cancer work.
In many ways, yes. That massive effort, born out of crisis, truly paved the way for the advancements we're seeing now in oncology.
Yeah.
It solved fundamental problems that were holding back the cancer applications, too.
That's fascinating how one challenge helped accelerate solutions for another. So now that we're seeing these technical bottlenecks largely overcome, what kinds of mRNA cancer vaccines are currently in development? Where are they in clinical trials?
Okay, so there are a few promising types, each tailored to different aspects of cancer. One significant approach involves targeting cancers that are known to be associated with viruses.
Like HPV.
Exactly. We're talking about human papillomavirus, HPV, hepatitis B, HBV, Epstein-Barr virus, EBV, and even HIV, which can contribute to certain cancers. For these, mRNA offers a therapeutic approach, not just preventative like traditional HPV vaccines. It aims to treat existing infections or associated cancers.
So treating, not just preventing.
Right. For example, for HIV, an mRNA-based vaccine has induced both humoral, that's antibody responses, and cellular immune responses in mice and primates.
Promising results in animals.
Yes. Rhesus monkeys, in one study, showed an impressive 79% reduction in SHIV exposure. That's a model virus for HIV. And very recently, a phase I clinical trial against HIV showed good safety and induced broad neutralizing antibodies in 97% of participants. That's a very encouraging early sign for human application.
97% way. That's incredible progress for virally linked cancers. But what about cancers that aren't linked to viruses? What's the strategy there?
For those, scientists are focusing on what we call tumor-associated antigens, or TAAs.
TAAs. Okay.
These are proteins that are expressed at significantly higher levels in tumor cells compared to normal healthy tissues. They might be present in normal cells in small amounts, but their overexpression in cancer makes them prime targets for the immune system.
So the immune system can tell the difference by the quantity.
Pretty much. An excellent example is BNT111. That's an mRNA vaccine targeting 4 common TAAs found in melanoma, serous skin cancer.
Melanoma, right.
In a phase I trial, it induced new or enhanced pre-existing immune responses in over 90% of patients with cutaneous melanoma. And now a phase II trial, combining it with samiplimab, another immunotherapy drug, is actively underway.
Okay, that sounds promising. And then there's the most personalized approach I've heard of targeting something even more specific than TAAs. Can you explain that?
Yes, that would be targeting tumor-specific antigens, or TSAs, which are also known as neoantigens.
Neoantigens. Okay.
These are truly unique. They are proteins or peptide fragments that are either uniquely expressed or significantly overexpressed only in cancer cells, often due to specific mutations within that individual's tumor DNA.
So they're not found in healthy cells at all.
Crucially, yes, they're generally absent in normal, healthy tissues. This makes them absolutely ideal targets for the immune system because there's minimal risk of the immune system attacking healthy cells and the off-target effects.
Much more precise.
Exactly. These personalized mRNA neoantigen vaccines are identified through incredibly advanced sequencing and bioinformatics approaches that analyze a patient's individual tumor genetic makeup.
Tailor-made for each person's cancer.
That's the goal. For instance, mRNA 4157, when this was combined with pembrolizumab, a common checkpoint inhibitor immunotherapy for high-risk melanoma patients, get this, it dramatically reduced the risk of recurrence or death by 44% and 49% respectively in a phase I trial.
Reduced risk by almost half. That's monumental.
It really is. And this marks mRNA 4157 as the first personalized mRNA cancer vaccine to reach phase 3 clinical trials, which is that critical large-scale stage before potential approval.
So it's getting closer to actual use.
It is. And we're also seeing similar personalized approaches being developed, like BNT122 for pancreatic cancer, which is another highly challenging malignancy known for being difficult to treat.
That truly sounds like the future of precision medicine in oncology. It's incredible. And finally, I understand mRNA can also be used to kind of remodel the environment around the tumor itself, not just target the cells directly.
Precisely, and this is another really innovative application. These are called mRNA-encoded immunomodulators.
Immunomodulators, okay.
Imagine a fortress, right? A tumor often creates its own defensive microenvironment, the immunosuppressive tumor microenvironment, or TME.
Like a shield.
Exactly, like an invisible shield. This TME actively suppresses the immune response that wants to fight the cancer. It tells the immune cells to back off or shuts them down.
So the body's army can't get in.
Right. mRNA-encoded immunomodulators are designed to be like highly specialized demolition teams. They deliver instructions for making things like cytokines, co-stimulating ligands, or other immune regulatory factors directly into this hostile environment.
To break down the defenses.
Exactly. The aim is to remodel that TME, break down those defenses, and make the tumor more visible and vulnerable to the immune system or to other immunotherapies that the patient might be receiving.
So it helps other treatments work better too.
Potentially, yes. It helps enhance the tumor's sensitivity. An example is delivering mRNA instructions for making the p53 protein, a well-known tumor suppressor, directly to liver cancer cells. When combined with other drugs like anti-PD-1 antibodies, it showed effectiveness in modulating the TME and achieving anti-tumor effects in early studies.
Fascinating.
Another example, mRNA 2752 encodes 3 different immune regulatory factors and is currently in phase I and phase II clinical trials for various advanced malignancies. It really showcases the incredible breadth and versatility of mRNA's potential tackling cancer from multiple angles simultaneously.
The scientific ingenuity we've just discussed is nothing short of revolutionary. I mean, it's just staggering. But the true impact, and indeed the most complex questions, only emerge when these breakthroughs intersect with the unpredictable landscape of human society, our individual rights, and our collective responsibilities.
Yeah, that's where it gets really complicated.
It's a constant negotiation, isn't it, between what we can do scientifically and what we should do ethically and strategically for public health. Let's now explore the human element, ethics and strategy in public health. And here, we immediately encounter that classic balancing act, individual autonomy versus collective well-being.
That's a crucial insight. Vaccination, fundamentally, stands as a cornerstone of public health. It offers some of the most potent tools we have to prevent and control infectious diseases on a population scale.
Right, it's about protecting the community.
But at its core, any public health intervention, particularly one as widespread as vaccination, is guided by fundamental ethical principles. We're talking about autonomy, the individual's right to make their own choices.
Self-determination.
Exactly. Then there's beneficence, the duty to do good and maximize benefits, minimize harm. And justice, which demands fair and equitable distribution of benefits and burdens across society.
And these principles can clash.
They often do. They're frequently in tension, creating really complex dilemmas, especially with vaccines.
Autonomy makes perfect sense here. It's about a person's right to choose, to make their own decisions about their body and their health without coercion. In the context of vaccines, this brings up the whole idea of informed consent.
Exactly. Informed consent is a cornerstone of ethical medical practice. It ensures individuals make truly voluntary and knowledgeable decisions about vaccination.
Knowledgeable being the key word there.
Absolutely. This means providing comprehensive, understandable information about the benefits of a vaccine, its potential risks and side effects, and any available alternatives. However, this process becomes incredibly complex in the face of vaccine hesitancy.
Yeah, that's a big one.
That whole spectrum of reluctance or refusal to vaccinate, despite the availability of vaccine services. Overcoming hesitancy necessitates transparent communication, accessible information that addresses concerns directly and respectfully, and open, empathetic dialogue, not just one-way directives.
It's about building trust, really.
Absolutely. Trust is foundational, and it takes time and consistent effort to build and maintain.
And then there's the really thorny issue, the debate over vaccine mandates. That really pushes the balance point between individual choice and the greater good of the community. Ethically, how do we even begin to navigate that?
This is genuinely one of the most contentious ethical dilemmas in public health. Vaccine mandates, while they align strongly with the collective goal of achieving herd immunity and protecting vulnerable populations.
Which is a clear public health benefit.
Undeniably. But mandates also undeniably raise significant ethical considerations regarding individual autonomy. They essentially compel individuals to vaccinate, which can be seen as infringing on their freedom to make personal healthcare choices.
Right, the my body, my choice argument often comes up.
Exactly. The challenge lies in striking a justifiable balance between those public health imperatives, the need to protect the community from serious infectious diseases, and individual autonomy. When mandates are considered, ethical frameworks generally suggest they should be proportionate to the public health threat, they need to be demonstrably effective, and ideally, they should include provisions for medical exemptions and, in some contexts, philosophical or religious exemptions too.
So acknowledging diverse circumstances and beliefs.
Right. It's about carefully weighing the harm prevented for the many against the burden placed on the few, and ensuring it's the least restrictive means necessary to achieve the public health goal. It's a tough calculation.
Beyond personal choice, there's also the very practical side, the economics of prevention versus treatment. It's a long-running debate in healthcare, isn't it? Is it better financially and societally to invest upfront to prevent a disease or wait and treat it once it develops?
It's a crucial question, with monumental implications for healthcare systems globally. Look, from a purely economic standpoint, preventing a disease from ever taking hold is almost always going to be more favorable than waiting for it to develop and then trying to treat it, right?
Seems logical.
As Benjamin Franklin wisely put it centuries ago, an ounce of prevention is worth a pound of cure. Saves money, saves suffering.
Go ahead.
But the significant hurdle is that prevention almost always incurs an upfront cost. Whether that's for vaccine development, massive public health campaigns, or screening programs, you have to spend money now for a benefit later.
The return on investment isn't immediate.
Exactly. And this creates a challenging dynamic, especially in multi-payer healthcare systems like we have in the USA. The entity investing in prevention, say, a specific insurance company providing vaccines, doesn't always reap the long-term financial benefits because patients might switch insurance providers or healthcare systems before that preventative investment fully pays off down the line.
So there's a disconnect between who pays now and who saves later.
Precisely. It's a structural challenge. Who captures the value?
That's a fascinating point about the payer models. For really expensive preventative therapies or maybe one-time treatments, which we're seeing more of with things like gene therapies, how do healthcare systems or pharmaceutical companies try to address this upfront cost issue? Are there creative solutions emerging?
This is where innovative thinking is truly needed, and we are seeing some movement. What's fascinating here is the emergence of novel payer models, especially for incredibly expensive, often one-time gene therapies that are essentially preventative in nature. They prevent a lifetime of chronic illness.
Like fixing the problem at the source.
Right. Novartis, for example, introduced an annuity-based model for its spinal muscular atrophy, SMA gene therapy, Zolgensma. This is one of the most expensive drugs ever brought to market, millions of dollars per dose.
Wow. How did the annuity model work?
Instead of demanding an upfront lump sum that could break a hospital or insurer's budget, they worked with payers to create 5-year outcomes-based agreements and novel pay-over-time options.
Outcomes-based, so payment depended on it working.
In part, yes, and spreading the cost out. It recognized that one dose of Zolgensma is designed to replace a lifetime of incredibly expensive chronic therapy for SMA. While it's not a vaccine, it serves as a powerful example of the kind of innovative financial models that can be explored to make prevention economically viable for healthcare systems. It bridges that gap between the high upfront investment and the massive long-term societal and economic benefit.
So creative financing to match the long-term value.
Exactly, acknowledging the immense value of a single intervention that can avert decades of chronic care costs and improve lives dramatically.
And it's not just about the science or the economics. Social dynamics play a huge role in whether people actually adopt preventative health measures. It's a classic human challenge, isn't it?
Oh, absolutely.
We all know we should exercise more or eat healthier, but actually changing behavior, even with the best intentions and clear benefits, can be incredibly tough. Vaccines face that same behavioral hurdle.
It truly is a fundamental aspect of public health, maybe one of the hardest parts. People generally want to do preventative things like exercise and eat healthy, and they understand the general benefits. We all do.
Sure.
But genuine, sustained behavior change often only comes when it is reinforced on a variety of fronts through clear messaging, seeing it as a social norm, having easy access, and critically, trust in the system providing it.
It needs multiple layers of support.
Definitely. We've seen this incredibly well with HIV treatment, for instance. Think about how that evolved. It went from public awareness campaigns in the late 1980s that focused purely on prevention through education.
Right, condoms, awareness.
To an expectation of effective disease management using antiretroviral therapy in the mid-90s and beyond. That transformed HIV from what was seen as a death sentence into a manageable chronic condition for many.
Huge shift.
And today there's even pre-exposure prophylaxis, or pre-OK, with drugs like Truvada. This enables HIV-negative people at high risk to protect themselves from infection in the first place. This whole evolution, from just awareness to treatment to robust prevention, demonstrates a real willingness by individuals to accept and adhere to preventative therapies when the value, necessity, and effectiveness are clearly established and reinforced within society.
That's a great example of long-term change. It brings us to how we actually talk about vaccines and how we combat the seemingly constant spread of misinformation. It feels like an uphill battle sometimes, especially in the age of social media where narratives can spread like wildfire.
It definitely feels that way.
So what's the strategic approach to communication in this really complex environment? How do public health bodies navigate this?
Well, at its core, transparency is absolutely paramount. Effective communication strategies for vaccination require clear, honest, and accessible information. It needs to be presented without jargon or condescension.
Talk to people like adults.
Exactly. This includes openly discussing the vaccine development process, thoroughly explaining safety profiles, and acknowledging potential side effects. And crucially, being upfront about uncertainties or remaining questions. Don't pretend you know everything if you don't.
Honesty builds trust.
Precisely. Establishing and maintaining trust is arguably the single most important factor in overcoming skepticism and building confidence in vaccination programs. If people feel like information is being withheld or spun, that trust erodes very, very quickly.
So if transparency is the cornerstone, how do you address vaccine hesitancy head-on when you're actively trying to build that trust, especially when there's just so much conflicting information flying around online and elsewhere?
Strategic communication needs to address these concerns directly and, importantly, empathetically. It's not about dismissing concerns, even if they seem unfounded.
Listen first.
Right. Public health campaigns need to leverage clear, concise messaging to not only debunk myths, but also correct misinformation by providing accurate, evidence-based alternatives. It's not enough to just say something is false. You need to explain why and provide the correct context.
Give people the tools to understand.
Yes. Emphasizing the safety and efficacy of vaccines through credible sources is crucial. But perhaps most importantly, tailoring messages to specific populations is key. Different communities have different cultural beliefs, varying levels of scientific literacy, unique historical experiences with healthcare, all of which influence their vaccine attitudes.
So no one-size-fits-all message.
Absolutely not. A generic campaign simply doesn't work for everyone. You need to understand the audience.
And what about social media? We often hear how it's just this, breeding ground for vaccine misinformation, with public figures often seen as driving those narratives. It paints a picture of a direct, almost linear spread of harmful content from influential people.
Yeah, that's the common narrative.
But a recent Twitter analysis, or X, I guess we call it now, provided a truly unexpected insight, didn't it? It seems to challenge a lot of our assumptions about who influences whom online and how anti-vax content actually functions in those spaces.
That analysis was genuinely eye-opening. Yeah, a huge aha moment for understanding the complexities of online discourse around this topic. What the researchers found was pretty surprising. Most discussions surrounding public figures and common hashtags labeled as anti-vax actually did not reflect genuine anti-vaccination beliefs from the people posting.
Really? So what were they reflecting?
Only about 2% of these messages included direct anti-vaccination content. Instead, the study concluded something quite different. Accusing public figures of holding anti-vaccination attitudes was often being used as a means of insulting and discrediting the public figure themselves.
Oh.
Rather than being an active attempt to discredit vaccines, it was more about political or personal attacks using the anti-vax label.
So the label itself became a weapon.
Exactly. The vast majority of these messages, in fact 61% of them, were actually condemning public figures for expressing perceived anti-vax beliefs, not promoting them.
So if I'm understanding this correctly, it's less about celebrities directly convincing people to be anti-vax in these specific online fights, and more about people using the anti-vax label as a tool, an insult in other political or social debates, often to condemn those views.
That's what the study strongly suggests, yes. It's a critical distinction.
That's a powerful distinction and a truly surprising insight into how online conversations around health can be co-opted for broader social dynamics. It definitely shifts the focus from just the vaccine content itself to the social and political dynamics around it.
Right. It points to a much more complex information ecosystem than a simple one-way outsized influence of celebrities on vaccination attitudes.
Who are some of these public figures caught in this crossfire, according to the study?
Well, public figures mentioned prominently included the tennis star Novak Djokovic. He apparently faced a significant amount of ridicule, with 61% of the insults directed specifically at him in that context.
Wow, okay.
Interestingly, political figures like Kamala Harris, Joe Biden, and Donald Trump were also part of this discourse. Trump, for instance, was noted as accusing Biden and Harris of having anti-vax beliefs.
So it became part of the political sparring.
Exactly. This highlights how such accusations are wielded not necessarily to convert people to an anti-vaccine stance in those threads, but as a political tool or a way to discredit opponents in public discourse. It reveals that the term anti-vax itself has become a loaded label, used to signify a certain position within a broader cultural or political divide.
So given this complex landscape where the conversation isn't always what it seems on the surface, what's the crucial takeaway for public awareness campaigns? How do we communicate effectively when the battlefield is so nuanced and messy?
Well, it absolutely underscores the critical need for culturally competent campaigns.
Culturally competent.
Meaning messages must be meticulously adapted to align with the specific cultural beliefs, values, and practices of diverse communities. This isn't just about translating language. It's about understanding the deep nuances of how health information is received and trusted within different groups.
Going beyond the surface level.
Exactly. It involves actively collaborating with local community leaders, religious figures, cultural influencers, people who already hold the trust of their communities. By doing so, public health communicators can ensure that vaccination information is not only scientifically accurate, but also culturally sensitive and respectfully presented.
So working with communities, not just talking at them.
Precisely. Fostering trust and encouraging informed decision-making from within the community itself, rather than trying to impose it from the outside. That's likely to be much more effective in this complex environment.
Okay, so we've explored the incredible science and the intricate ethical and communication foundations, but now let's talk about the bigger picture. Vaccines in the public sphere, perception and politics. This is where high-level policy decisions, political agendas, and global dynamics really come into play, shaping how these vital tools are actually used and accessed.
Absolutely. This is where the rubber meets the road, so to speak, in terms of large-scale impact. Politicians are frequently the high-level policymakers in healthcare, right? And their fundamental attitudes towards prevention versus treatment profoundly shape the policies that ultimately emerge.
Their personal beliefs or party lines can have real consequences.
Definitely. We saw a stark, real-world example of this influence with a client's diagnostic product a while back. This product enabled early detection and prevention of a serious condition.
Okay.
Analysis showed that Democratic states appeared to embrace these kinds of tools for prevention and early detection much more readily, while Republican states adopted them significantly less so.
A clear political divide in uptake.
A very clear divide. This directly impacted patient access and resource deployment, illustrating the significant and tangible influence political agendas can have on public health policy, sometimes seemingly independent of the scientific consensus or evidence.
And it's not just politicians shaping things. The public itself can be hesitant, even with long-established preventative measures that have proven benefits over decades. It's not a new phenomenon tied just to recent vaccines, is it?
Not at all, no.
No.
Even with increasing public and scientific support for prevention generally, there are still many voices raising doubts around risk-benefit profiles. And there's often a genuine, sometimes deeply held fear of unintended long-term consequences, even for things we think are settled science.
Like what, for example?
Well, historically, we've seen concerns and public debate around things like routine cervical cancer screening or the measles, mumps, and rubella MMR vaccination program. These aren't new controversies. They've been subject to scrutiny and public debate for decades, sometimes flaring up again and again.
So this kind of skepticism isn't unprecedented.
Not at all. This persistent skepticism highlights the ongoing need for public health advocates and policymakers to constantly keep prevention at the very top of the political agenda, to continually drive awareness, transparently address concerns as they arise, and work to maintain high adherence through sustained public trust and education. It's never a done deal.
What's fascinating here is how these political attitudes and even that long-standing public skepticism can translate into concrete governmental decisions, sometimes with immediate and pretty sweeping consequences. We've seen some significant headlines recently that really illustrate this point, haven't we?
We certainly have. It's been quite prominent. For example, the current U.S. Health Secretary, Robert F. Kennedy Jr., who is known for being a longtime vaccine critic.
Right, that appointment made waves.
He notably made a decision to cancel $500 million in government-funded research projects. These projects were specifically aimed at creating new mRNA vaccines against respiratory illnesses with the goal of being better prepared for future health emergencies, future pandemics.
$500 million in research canceled.
Yes. And this move has, understandably, been met with considerable dismay by many infectious disease experts. They argue that rigorous, proactive science, the kind needed for preparedness, is being abandoned under his leadership.
And there were other changes, too.
Concurrently, there was also the departure of a somewhat controversial FDA vaccine chief, which some observers viewed as part of this broader shift in approach within health agencies. These incidents really highlight the direct and significant impact that political appointments and specific political perspectives can have on crucial public health policy, scientific research funding, and the overall direction of national health preparedness. It's a stark reminder.
This brings us to a global challenge that really underscores that principle of justice we talked about: global equity and vaccine diplomacy. It's one thing to develop these incredible vaccines, but quite another to ensure they actually reach everyone who needs them all around the world. The pandemic really laid bare these disparities, didn't it?
Oh, absolutely. Brutally so. The grim reality is that access to vaccines remains a profound challenge in many developing nations. It starkly highlights ethical concerns related to justice and equity on a global scale.
What did we see during COVID?
During the COVID-19 pandemic, we saw wealthy nations very quickly secure vast quantities of vaccines through advanced purchase agreements, often far exceeding their immediate needs.
Vaccine hoarding, some called it.
Yeah. While at the same time, many low-income countries struggled desperately to inoculate even their frontline healthcare workers and most vulnerable populations. This created what some commentators starkly termed vaccine apartheid.
That's a strong term.
It reflected a harsh reality. The ethical imperative here is crystal clear. Ensuring equitable access to vaccines underscores the urgent need to address these entrenched global health disparities. Resource allocation, everything from manufacturing capacity to funding to actual distribution logistics, should prioritize the most vulnerable populations, regardless of their geographical location or economic standing. It's really about a global moral obligation.
So, how do we begin to address this on an international scale? It seems like such a huge systemic problem. What kind of collaboration and global initiatives are needed to try and bridge these massive gaps?
International collaboration and solidarity are not just desirable, they are absolutely essential. There's no other way. Initiatives like COVAX, for instance.
Right. I remember COVAX.
COVAX was established with the ambitious aim to pool resources and provide equitable access to COVID-19 vaccines for low and middle-income countries. Its goals directly aligned with the principles of justice and fairness.
But it faced problems.
It did. COVAX also faced significant challenges, including rampant vaccine nationalism, where countries understandably prioritize their own populations first, sometimes breaking prior commitments. And there were huge logistical hurdles in last-mile delivery in resource-poor settings.
So good intentions weren't enough.
These experiences really underscore the need for genuine solidarity and resource allocation. fostering alliances that truly prioritize global well-being over narrow geopolitical competition or national interests. It's about building a truly collective response capacity, not just scrambling during a crisis, but as an ongoing commitment to global health security.
And then there's this term vaccine diplomacy, which emerged quite prominently during the pandemic. It sounds like it could potentially be a force for good, sharing resources, but it also carries its own set of ethical complexities, doesn't it?
It absolutely does. It's a double-edged sword. Vaccine diplomacy refers to the strategic use of vaccines, donating them, selling them on favorable terms as a tool for international influence, for enhancing geopolitical standing or soft power.
Using vaccines to make friends or gain leverage?
Essentially, yes. Now, while nations certainly have a primary duty to secure the health and well-being of their own populations, the inherently global nature of infectious diseases, they don't respect borders, demands extensive collaboration. This involves navigating really complex ethical dilemmas.
Like what?
Like, how do nations balance their own national interests and domestic needs with their responsibility to contribute to global health? The real danger is when vaccine distribution becomes primarily a bargaining chip, used to extract political or economic concessions, rather than being driven primarily by humanitarian concerns and public health needs.
So, aid with strings attached.
Potentially. Ideally, collaborative efforts should be underpinned by a genuine commitment to shared well-being, fostering alliances that truly prioritize global solidarity over geopolitical competition. We need to ensure that humanitarian aid, including vaccine sharing, is not merely a thinly veiled form of influence peddling. It's a fine line to walk.
Now let's peer into the future. What's on the horizon of vaccination? It feels like with all the breakthroughs we've discussed, especially with RNA, we're maybe just scratching the surface of what's possible, not just in the lab with the vaccines themselves, but also in how they're actually delivered to people.
We truly are. It's a very exciting time. Emerging technologies are reshaping the future of vaccine development and delivery in profound ways, moving us well beyond even the linear mRNA we've spent a lot of time discussing. The innovation pipeline is incredibly rich right now.
Okay, tell us about some of these new RNA vectors then. Beyond the standard mRNA that we saw with COVID, what else is exciting researchers in the RNA space?
Well, one really promising vector is self-amplifying RNA, or SARNA.
Self-amplifying, meaning it makes copies of itself.
Exactly. Think of traditional mRNA as like a single-use instruction manual. Your cell reads it once to make the protein, and then the mRNA degrades. Now imagine SARNA as a special instruction manual that, once delivered into your cells, can actually make copies of itself over and over.
How does it do that?
It's usually derived from a type of virus, and it carries its own little copying machine, an enzyme sequence called an RNA polymerase, embedded within it. Because it keeps churning out those protein-making instructions, it exhibits self-amplification and translation capabilities.
So you need less of it to start with.
Potentially, yes. You might need only minimal quantities to elicit a much more robust and detectable protective immune response. Studies have shown SARNA targeting human papillomavirus, for example, can exhibit stronger anti-tumor effects compared to standard non-replicating mRNAs.
Are there downsides?
There are still challenges. SARNA molecules are larger and more complex than standard mRNA, which can make delivery harder. And there's still some question about whether that embedded RNA polymerase copying machine might trigger an unintended immune response itself in humans. Researchers are actively working on addressing those issues.
Okay, that's clever, boosting the signal. And there's also circular RNA, right? circRNA. That sounds like it would have some unique benefits just based on its shape.
Indeed. Circular RNA, or circRNA, is another fascinating development. Unlike linear mRNA, which has open ends like a piece of string, circRNA has a covalent circular structure. It's a closed loop.
Like a bracelet versus a necklace.
Good analogy. This circularity offers significantly enhanced stability over linear mRNA. Why? Mainly because it's highly resistant to certain RNase enzymes. Those are the body's natural scissors that normally chew up RNA from the ends inwards.
So it protects it.
Right. This characteristic provides distinct advantages for RNA vaccines. It could potentially lead to longer-lasting antigen production from each molecule, and therefore stronger immune responses, possibly with lower doses needed.
Any evidence for that yet?
Yes, studies have shown circRNA vaccines protecting against multiple COVID-19 variants. And in some cancer models, they've even shown more effectiveness in tumor suppression than linear mRNA vaccines. Sometimes they can even act as their own immune adjuvant, sort of boosting the overall immune response.
So more stable, potentially more potent. What are the hurdles there?
The main challenges right now are scaling up the synthesis efficiency, making large amounts of it purely and correctly, and managing the cost of the specific regions needed to make those circular structures. It's still a newer technology in terms of manufacturing.
Okay. What about something completely different in terms of RNA structure? I think I heard about something called comb-structured RNA. That sounds unusual.
It does sound unusual. That's a very cutting-edge concept, really pushing the boundaries. A recent advancement involves hybridizing short pieces of double-stranded RNA, or dsRNA, onto a backbone of single-stranded mRNA. This creates a unique dendritic or comb-like structure.
Like teeth on a comb.
Exactly.
Mm.
Now, the genius here is that this structure is designed to act as both an antigen expression vector, carrying the instructions for the immune target, like the mRNA backbone does, and as an immune adjuvant simultaneously. Adjuvants, remember, are substances that enhance the immune system's overall response to a vaccine.
So it carries the message and rings the alarm bell louder.
Precisely. This clever design allows for potentially much more precise control over the strength and pipe of the immune stimulation. The hope is that this could significantly enhance both vaccine efficacy and safety by allowing us to fine-tune the immune response much more accurately than before. It's an elegant solution combining multiple functions into a single, highly structured molecule. Very early days, but very exciting.
Wow. Okay, so we have these incredible new RNA technologies coming online, constantly refining the actual blueprint of a vaccine. But what about the delivery? For many people, let's be honest, the thought of needles is still a significant deterrent.
Absolutely. Needle phobia is real and widespread.
Are there innovations happening there that could finally move us beyond the needle?
Yes, absolutely. The need to move beyond the needle is a huge driver of research, driven both by patient comfort and, crucially, by the need for better global access. Needles can be intimidating, they require trained personnel for administration, and they generate sharps waste, which is an environmental and safety issue.
Right, disposal is a problem.
So scientists are actively exploring lots of innovative ways to administer vaccines that are potentially cheaper, easier, less invasive, and could even allow for self-administration or reach into remote areas more easily.
Like oral vaccines. I remember the oral polio vaccine being a massive game changer back in the 1960s, helping to nearly eradicate polio.
Exactly. Oral vaccines are already in use, as you mentioned, with the Sabin oral polio vaccine, and they remain very promising. The potential advantages are huge. They're often cheap to produce, incredibly easy to administer, just swallow, and they can be extremely effective, often eliciting strong mucosal immunity right in the gut, which is where many pathogens first try to enter the body.
So what's the catch? Why aren't all vaccines oral?
The main challenge, unfortunately, is our incredibly hostile digestive system. Stomach acids, digestive enzymes. They can easily damage or destroy sensitive vaccine components like proteins or RNA before they even have a chance to trigger an immune response further down in the intestines.
So the delivery vehicle needs serious protection.
Precisely. Researchers are now looking at sophisticated solutions like microscopic protective layers, special capsules, or even using harmless bacteria to carry the vaccine components, all designed to ensure the vaccine retains its effectiveness as it makes that perilous journey through the digestive tract.
And what about nasal sprays? They seem very user-friendly and would make a lot of sense for respiratory viruses like flu or COVID.
Nasal sprays offer a fantastic pathway, especially for respiratory pathogens. They have the significant advantage of not requiring specialized medical training for administration.
So no nurse needed?
Potentially, yes, meaning people could maybe even vaccinate themselves at home eventually. The nose itself is actually highly effective for vaccine delivery. It has a very dense network of blood vessels right near the surface, and it's lined with a porous membrane that allows for pretty efficient absorption of vaccine components into the bloodstream and lymphatic system.
Plus, it's where those viruses often enter.
Exactly. Since the nose is a common entry point for many viruses, nasal sprays can also directly boost the immune response right where it's most needed at the mucosal surfaces of the respiratory tract. This can be really important for blocking initial infection. A nasal spray vaccine for flu in children is already approved and used in the UEA, and there's extensive research actively ongoing for other respiratory viruses like COVID-19 and RSV using this route.
Okay, that sounds very practical. What are some other through-the-skin innovations beyond just a traditional jab with a needle?
Yeah, beyond standard injections, researchers are exploring painless or less painful delivery through the skin using a few intriguing methods. One approach is using jets of high-pressure air or gas.
Like a tiny pressure washer?
Kind of. It can push the liquid vaccine formulation through the outer layers of the skin without using a needle at all. Another method involves using focused ultrasound waves. These sound waves can temporarily increase the skin's permeability, making it easier for vaccine molecules to pass through.
Ultrasound. Interesting.
Both of these methods leverage the fact that the skin itself is an ideal place for many vaccines to interact with the immune system. It's absolutely packed with specialized antigen-presenting cells, like Langerhans cells and dendritic cells, which are crucial for initiating a strong immune response.
So delivering it right to the immune system's front door, are there challenges?
Yes. Challenges for these methods include ensuring a uniform depth of penetration every time, which can be tricky given that skin thickness varies across the body and between individuals. And currently, the devices needed for these methods tend to be more complex and costly than a simple syringe and needle.
Okay. Now, microneedles sound particularly intriguing. You mentioned tiny needles that don't actually hurt. That almost sounds too good to be true for people scared of needles.
That's precisely the idea, and a major driver for improving patient acceptance. Microneedles are typically patches covered with dozens or even hundreds of absolutely tiny needles. They're usually less than a millimeter long, shorter than the width of a human hair sometimes.
So incredibly small.
Yes, so short that they pierce only the very outermost layer of the skin, the stratum corneum, without reaching the deeper layers where the pain-sensing nerves and blood vessels are located.
So no pain, no blood.
Generally, yes. This technology effectively eliminates the visual fright factor of seeing a large needle, reducing anxiety, and crucially, causes little to no pain. These patches can even enhance vaccine efficacy in some studies because they deliver the vaccine very efficiently directly to those immune-rich superficial layers of the skin we just talked about.
Wow. And what was the other one? Electroporation. That sounds a bit sci-fi.
It does sound a bit futuristic. Electroporation is a different approach altogether.
Right.
This method involves applying a small, controlled, very brief electrical pulse to the skin or muscle tissue right after the vaccine is injected, sometimes with a special applicator.
Electricity. Why?
This small electrical field does a couple of things. It can help drive the vaccine molecules more efficiently into the body's tissues, but more importantly, it can temporarily make the membranes of nearby cells more permeable. It sort of briefly opens tiny pores in the cells. This enhances the uptake of the vaccine, particularly DNA or RNA vaccines, right into the cells where they need to work.
So it boosts the delivery efficiency using physics.
Exactly. It's about using physics to improve the biology of vaccine uptake and the subsequent immune response. It's being actively explored, especially for DNA vaccines and some therapeutic cancer vaccines.
This all sounds incredibly promising. It really points towards a future where vaccines could be much more accessible, far less intimidating, and perhaps even more effective. But with such rapid technological advancement happening on so many fronts, there must be new ethical challenges emerging alongside them, right? We can't just forge ahead technologically without carefully considering the profound implications of these innovations for individuals and society.
You've hit on a critical point. Absolutely. These technological advancements inevitably bring new challenges and anticipatory ethics for the future. We have to think ahead.
Like what kind of new dilemmas?
For instance, consider those personalized vaccines we talked about, tailored to an individual's unique genetic makeup, like the neoantigen cancer vaccines. While incredibly powerful medically,
They raise other questions.
big questions. Complex ethical concerns regarding potential genetic discrimination. Could this highly personal genetic information derived from your tumor somehow be used against you, perhaps by insurers or employers? There are also paramount issues of data security breaches. How do we protect this incredibly sensitive genetic data?
And access too, presumably.
Yes, and the huge question of unequal access. If these personalized therapies are extremely expensive to develop and administer, who will actually have access to them? Will this just exacerbate existing health inequities between the rich and poor or between different countries?
Right. And what about AI? Artificial intelligence is being used more and more in drug development. Does that bring ethical issues too?
It does. The growing use of artificial intelligence in vaccine development, everything from identifying potential viral targets to optimizing vaccine formulations or predicting pandemic spread, brings its own set of issues. Transparency is a big one. How do we know how the AI reached its conclusions or recommendations? Can we trust it? Accountability is another. If an AI-designed vaccine leads to unforeseen problems, who is responsible? And there's the significant potential for bias embedded in the algorithms of the training data used to teach the AI, which could lead to vaccines that work less well for certain populations.
Lots to consider there. And what about ethics in rapid response situations, like during a pandemic when speed is just absolutely paramount? The discussions around emergency use authorizations or EUAs during COVID-19 were particularly intense.
They absolutely were, and they highlight really significant ethical challenges inherent in crisis response.
Right.
EUAs allow for the rapid deployment of vaccines or treatments without the full, extensive, multi-year testing typically required for full regulatory approval.
Which is necessary for speed.
Vital in an emergency, yes. But this process raises fundamental questions about obtaining truly informed consent when the full long-term safety and efficacy data might not yet be available and when public anxiety and pressure are incredibly high.
How can consent be fully informed if the information is incomplete?
Exactly. It requires a very delicate balance. It demands exceptionally transparent communication about what is known and, just as importantly, what is unknown at that point. It requires robust risk communication that avoids both unnecessary alarmism and false reassurance. And ideally, it should involve establishing clear mechanisms for ongoing consent as more data becomes available post-authorization. We have to learn from the lessons of the recent past to refine these processes for the next inevitable crisis.
So with all these technological shifts, the personalization, the AI, the emergency use, what does this all mean for future ethical frameworks? How do we try to ensure these incredible scientific advancements are used responsibly and for the good of all, not just a select few?
The future undeniably demands what ethicists are increasingly calling anticipatory ethics.
Anticipatory ethics, meaning thinking ahead.
Precisely. It means that ethical considerations shouldn't just reactively follow scientific developments. They should, as much as possible, precede them or develop in parallel with the science. We need to proactively identify potential pitfalls, social disruptions, and ethical dilemmas before they become entrenched problems, and use those insights to guide research priorities and implementation strategies from the outset.
What should future frameworks prioritize?
Future ethical frameworks absolutely must prioritize justice and equity globally. We need mechanisms to ensure that new technologies benefit everyone, not just those in wealthy nations who can afford them. We need to leverage digital technologies thoughtfully to enhance informed consent processes, making complex information genuinely accessible and interactive for diverse populations. And critically, we must establish transparent and accountable global governance mechanisms for vaccine research, development, distribution, and data sharing, ensuring that the benefits of innovation are broadly and fairly shared across humanity.
That's a tall order, but clearly essential. And finally, let's consider something we don't immediately think of when we talk about vaccines, but which is increasingly vital in all aspects of our lives, the environmental impact. It's actually part of that bigger picture of prevention versus treatment, isn't it?
It absolutely is, and it's an increasingly important consideration that's starting to get more attention in healthcare sustainability discussions. From an environmental perspective, preventing disease, rather than perpetually treating it, will always be beneficial.
How so?
Well, think about it. The long-term treatment of many chronic diseases often demands a significant and continuous volume of drugs. Each of those drugs has an environmental footprint associated with its manufacturing processes, energy use, water use, chemical waste, plus raw material extraction, packaging, and global distribution logistics, often involving cold chains, which are energy intensive.
Right, the whole supply chain.
Exactly. And treatment also entails considerable consumption of hospital resources, energy for running facilities, disposal of vast amounts of medical waste, ongoing use of diagnostics, and other supplies. By preventing a disease in the first place through effective vaccination or other preventative measures, we inherently reduce this entire environmental footprint over a patient's potential lifetime.
So less disease means less resource consumption overall.
Exactly. So a successful vaccine or preventative therapy is not just good for individual health and potentially national economic budgets, it's also a powerful, though often overlooked, tool for environmental sustainability within the healthcare sector itself. It aligns health goals with environmental goals.
What an incredible deep dive we've had today. Seriously. We've journeyed from the truly revolutionary science of mRNA vaccines, exploring their mind-blowing potential not just for infectious diseases, but potentially for a whole new epoch in cancer treatment.
Yeah, the science is moving so fast.
Yeah, to the complex ethical tightrope walk between individual autonomy and collective well-being, something societies constantly grapple with. We've seen how public perception and political agendas can profoundly shape policy, sometimes in surprising ways. And we've glimpsed the exciting yet clearly challenging horizon of future vaccine technologies and delivery methods, promising perhaps a world beyond the needle for many.
It's definitely clear that the future of vaccination is not some predetermined trajectory. Nor is it solely the domain of scientists working in labs. It really is a collective responsibility. It's shaped continuously by ethical considerations, by strategic acumen in public health, by political will, and by the need for global collaboration.
Mm.
The lessons learned, especially recently, the breakthroughs achieved, and the challenges that still lie ahead, they all point to an ongoing journey that touches every single aspect of our lives and really underscores the absolutely vital role of preventative health and building a more resilient future.
So, as technology continues to push the boundaries of what's possible in health, from truly personalized vaccines tailored to our genes to maybe painless patches or nasal sprays replacing needles, here's a final thought for you, our listener. How do you envision our role, both individually and globally, in shaping an ethically sound and truly equitable future where the incredible promise of prevention actually reaches everyone?